Yes, good day, class. I welcome you today to our part B of revision of September paper 2, preparatory examinations. So what is posted on our board today is question 2, specifically question 2. I just managed to break down the presentations into pieces so that we can be able to have not boring time following up until to the end of the revision video that will be generated at the end of the presentation. So in this section, you are only going to find question 2, and please look forward to your question 3 that is just going to follow after this one as a new video, as a new presentation. So today we are working on question 2 specifically, just to make the videos short, not too long. So thank you so much for watching, and without wasting time, let us just go quickly to our, let's just go quickly to our presentations, to the questions. So note that these questions, they are actually like they're actually going to be analyzed and critically presented to you for the interest of what? Okay, very sorry for the technical glitch that just happened now. We can just continue. So let's just quickly go to our questions as fast as possible. We have got a question here that is question 2. Insulin is a peptide hormone produced by beta cells of the pancreatic islets encoded in humans by the gene. And then in most animals, including humans, a single gene for insulin is found. The human gene for insulin is located on the short arm of chromosome 11 at position chromosome 11 at position 5.5, 15.5. The diagram below represents a section of the human gene that codes for insulin. Strand A is a normal insulin strand, which is this one, strand A. This is our strand A class. Strand A is a normal insulin strand, and strand B is muted. Strand B is muted. So we are saying this is normal, and strand B is what? Is muted. It is a muted strand, and then this is what is shown there. Having the table below represents the key for the amino acids that represent, as represented in strand A and B. So we are having our structures being shown there. We have got a variety of amino acids that are given there, methionine, trison, proline, and so forth that are given here. These are our amino acids that are shown in the diagram. So if we can go to the next, if we can go to our questions are saying, according to the information given, what is the locus of human insulin? I'm sure that you remember what is the meaning of the word locus, the position of a gene on a chromosome. I'm sure that we still remember position of a gene on a chromosome is what we call a locus. So what is the position? What is the, according to the information given, what is the locus of the human insulin? Actually, this one, you can quickly just see the position is given there. It is given that the chromosome 11, the human gene for insulin is located on position, on position 11, on position what? On position 15.5. So it is actually given in class. The locus, which is asked in this case, is position what? It is position 11. Position 15.5. Position 15.5. 0.5, which is given on chromosome 11. I'm sure that we can agree together there on chromosome 11. It is given in the essay there. What type of mutation is illustrated in the diagram? What type of mutation is illustrated in the diagram? If we can check we can check on our diagram what type of mutation is illustrated. As you can see, we are saying this is a gene mutation that is given there. Diagram represented a section of a human instruction gene that codes for insulin. So we are looking at a gene, single gene for insulin is given there, single gene for insulin. So what type of mutation is shown? It is what? It is type of mutation that is illustrated is gene mutation. Question 2.21 is gene mutation class. This is gene mutation. Gene mutation. Identify the amino acid that is caused by mutation in strand B. Identify the amino acid that is caused by this, the type of mutation that is caused. In this regard, class, what you must understand is that you must see the difference in the structure of amino acids from the original to the muted one. So if we can check there, our amino acid strand A, which is normal, is this one. It has got its amino acids that are shown there, where we have got that A. I'm saying we can number this one 1, 1, 1, 1. It is the same as I was giving the ticks there, and then 2 and 2. You can see that they are showing they are the same. We are saying mutation is a change in the nucleotide basis that would result in a different in the formation of a different formation of a different amino acid. So you can see position 3 and position 3. We see there is this one is correct, but here we have got what? This is the muted one. It muted this one. So we have to look at what does this represent. It is representing what? What does it represent? We look on it and then find out that methionine is the one that is represented by this mutation. So it is methionine, which is represented by what? By the type of mutation that has taken place according to our diagram, where I'm saying on 3 and this one, we have a different amino acid that is formed here. So our question is saying what type, identify the amino acid that is caused by this mutation strand B. So if you look on it like I've illustrated, this results in what? In methionine. It results in methionine that you can see there. Even on our key is also written as what? Which is also written as met. It is also written as met on the key, on our key there of amino acid. And then the table below shows the amino acids and corresponding codes. It shows the amino acids. The table below shows the amino acid and their corresponding codes. This is the table amino acids and the codons there. I'm sure you remember in terms of the in regard to the types of RNA, to say where do we get codons and their anticodons. I'm sure that we can be able to pick up and say match them very well. 2.14, give the codon for the last amino acid in strand B. Give the codon for the last amino acid in strand B, the codon of the last amino acid in strand B. So we are given we have to go to our previous presentation, which is given that the strand B has got this, and then we must look on it and then check what is this amino acid that is represented by what? By listen. I'm saying it is, we have got this presentation or this key that represent the amino acid lysine, Lysine is the amino acid that is represented by the amino acid plant in strand B. Strand B, it has got this amino acid here class. So I'm saying we identify that it is representing lysine, and then in that respect, you go to the key, and then you see that lysine is that one. So you should go to the what? To the question now and check to say lysine is represented by which codon? This is lysine, and then the codon that is representing it is what? Is triple what? Adenine, which is given there. So the question is saying give the codon for the last amino acid in strand B as it is given here. This one, you don't calculate anything. It is given, which is what? Which is triple adenine, which is given there. It is actually given in this case. And then identify the DNA-based triplet for the third amino acid in the normal insulin. Identify the last amino. Identify the DNA. Identify DNA-based triplet for the third amino acid in the normal, and check here again in the normal strand, insulin strand. So we also refer back, or you must refer back to your what? To your diagram of the amino acid, which is what? Which is given in this regard. We are saying on the normal strand, this is our normal strand. Our normal strand is given. Our normal strand is what? Our normal strand, as I've indicated already, our normal strand is what? Is given as this one. So the question is saying, identify the question is want us to identify, it wants us to identify to say what is the, identify the DNA-based triplet for the third amino acid in the normal insulin strand. So what is the, identify the what? Identify the third amino acid DNA-based triplet for the, the third base triplet for the identify the DNA-based triplet for the third amino acid in the normal insulin strand. So the question is supposed to say you are supposed to identify the third amino acid, which is given as to say our third is marked here. This is our third amino acid that is being asked about. Our third amino acid that is asked about. I have already marked them like that to say what is the meaning of this one. What is the meaning of that? We go to our, this is our third, just to mark it again, our third, and then go and look what is that the diamond stand for. It is standing for what? For 309. So you come again to your question and check what is 309 representing. So in this case, we are given that this is what we are given to say this is the best triplet that we are referring to. So the best triplet that we are referring to is what? Is methionine. So hence, in this case, what I'm saying is you look on the third amino acid, which is this one, which has got this codon. So what the question is saying, you need to give the what? The DNA-based triplet. So it is gonna be what? If it is going to give you, that is going to be TG what? TGC. I'm saying the corresponding base triplet on this one, the DNA, it is supposed to be what? It's supposed to be TGC on this question, which you can give as say that is TGC class. That is what is given on this question specifically. I'm saying the base triplet of this one, the DNA-based triplet on this one, identify this one, and then identify the DNA-based triplet for the third amino acid in the normal insulin. So we are given this as the codon. So what we are saying is that what is the DNA-based triplet for that? It is supposed to be what? It is supposed to be TGC, which gave rise to the what? to say for us to have this mRNA codon, what was the corresponding base triplets there, DNA base triplets? It was TG, it was TGC, or it is TGC. That must give you AUG as the codon for this one. Thank you for getting it right. Let's go to the next question. Forensic scientists collected multiple hair samples at a crime scene in a family home. Our 4 family members live in the house. The diagram below shows the respective DNA profiles from the different hair samples found in the house. We have got the DNA base, the DNA profiles from the 4 different hair samples that we have found in the house. We have got sample 1, that is our sample 1, sample 2, sample 3, sample 4, and then sample 5 anyway. It shows that. So we are saying the question is therefore identify which hair sample could be that of the suspect. Identify which hair sample could be that of the suspect. If you can look here, we are saying, understand the question in the first instance. Forensic scientists collected 4 hair samples at a crime scene in a house, in a family home. 4 family members live in the house. The diagram below shows the respective DNA samples from the different hair samples found in the house there. So we are given as these ones. So in this regard, what you need to understand is to follow the question and don't make mistakes in any case. So the question says, identify which hair sample could be that of the suspect and explain your answer. So which one can be that of the suspect? It can be hair sample what? Hair sample, which one can be that of the suspect? Which can be that of the suspect. Look, if we look at them, we can take it to say that the one that could be of a suspect which looks different or which has got something that is unique, which we can refer to this, is going to be hair sample what? Hair sample 4. So it can be hair sample 4, hair sample 4 can be that of the suspect. And then now, why should we say that? Why should we say hair sample 4, 2.2.2? The reason is because none of the DNA profile bars of 4, if you can check there, none of the DNA profile bars, none of DNA profile bars of sample 4 match with any of the other samples profiles above. Match with what? Match with any of the what? Any of the samples that are above. Any of the samples. Any of the samples above. And then sample 4 is therefore not related in this case. Sample 4 is therefore not related to any of the people in there. Sample 4 is therefore not related to any of the people, any of the other people there, any of the other people there, which we are saying they're supposed to be 4. So if it is not related to any of the other people, hence it means this person does not live together with them. Hence does not does not live together with the 4 family members. With the 4 family members, hence is our prime suspect. So class, in these questions as these ones, you should not fail to get all of your marks, because when you are looking at them, like I'm saying, you can actually try to relate to say, if you can check sample 1, we have got this bar, we have got that bar, and then we have got this one, can also match to this one to say that they are related. We can also believe to say that this one and this one, they are relatives, which can also maybe mean to say this one can per se match to that. If you can look again, if you can look again, this one, this bar can match to that bar, which means to say that they are related. And then this one can also match to that, and can this match to this, which we can also come to this one, which is sample A4. Sample A4, this bar can match to this bar, which can also match to this bar. And then finally, we can also have a bar last matching to this bar, which we can conclusively say that these 1, 2, 3, and 5 are the 4 people who are staying together. They are staying together, stay together, stay together. This one is different, hence number 4 is our prime suspect. So class, just understand that when we are looking into this, we need to understand the matching of the bars, so therefore we can be able to relate them and give a possible suspect when questions such as these ones are to be asked. So please don't make mistakes. Don't make mistakes on this. And then the next question is saying, the possibility exists that the hair samples identified in question is not that of the suspect. Explain this statement. Explain the statement. Why that we can say that even if we can say number 4 can be the suspect, how can we not say that he is also the suspect based on some DNA scientific explanations? So in this case, how can we say that this hair cannot be that of the suspect? We got a hair sample in the house, but how do we explain that the person's hair or the hair sample that is found in the house is not gonna be that of the suspect? This question, class, it refers or it relates to a situation similar to the one that I once explained in my previous presentation, where I provided you with a scenario of a DNA sample that is found at a crime scene, and then explain why a certain person can be not fully convicted to say that, or cannot be conclusively say that the DNA sample is his. So the point is that in this case, we can also say that this question, there are possibilities that the hair could belong to anybody who visited the house. The hair sample, the hair sample, the hair sample can be for anybody who visited the house, class. It might happen that some friend has visited the house, and then after visiting, now there is a what? There is a burglary. So the hair sample may belong. It may belong to anyone who visited the house before. Belong to anyone who visited the house before, It might happen that somebody visited the house before. It can be, which can be what? Which can be a friend, which can be any other people, which can be a friend or any other people, or and then the presence of DNA does not prove that the person has committed crime before. Uh, who can be who can be a family friend, and and again in this case, and then the presence and the presence of the DNA sample in the house does not it does not prove that the person is a criminal, does not prove that the person is a criminal. It cannot prove that the person is a criminal class. That cannot be proven because of the DNA sample. So we say that it might happen that someone can visit the place before the crime scene. It might happen that the DNA sample might also have been planted by someone just to cause drama and cause problems on somebody. So it might happen that someone can plant the DNA in order to just implicate a family, I mean, some other people. Let's just answer our next question. The diagram below represents DNA's replication. Is a DNA replication that is shown in this question. This is a diagram of a DNA replication, which you can see that this diagram represents a nucleotide. This is a nucleotide represented there. And then we have got our strands, which I can nicely number to say strand 1. We can have our strand 1, and we can say also this is our strand 2, which just unzipped, and then the weak hydrogen bonds are broken, causing them to become what? These are our original strands. 1 and 2 are original strands, and then we are saying these also forms the templates. They are forming the templates of what? Of DNA replication. So without wasting time, give 2 visible reasons why this diagram represents DNA replication. Why does it represent DNA replication? Question 2.31. 2.31, why does it represent? How can you say this is DNA replication? 1, we are saying both strands act as templates. As you can see, I said strand 1 and 2. So give reasons why this can be, why this represents DNA replication. So reason one, both strands, we are saying both DNA strands act as templates. Both DNA strands act, they act as templates. Act as templates. That is first reason. So we are having DNA structure, and then it unwinds, having those strands acting as templates that I already mentioned, and I've given that to the class. And then we also have, in this case, we also have the matching of what? The matching of nitrogen bases. We can see nicely that there is G here, there is C there, there is T, and there is A. So there is A, there is T. So we can as well say that the nitrogen basis, what is it? Where we are seeing that thymine is attached to what? Thymine pays directly, it pays directly to adenine. That is given in our diagram there. We have got A, and T are pairing together, showing that it is specifically a DNA strand where we have got that matching nitrogen bases, and then adenine and thymine are found specifically on what? On DNA structure, and that is adenine and thymine pairing nicely there. And then two identical strands are formed, as we can see again. We have got we are two identical molecules. We have got two identical molecules, which I can show by giving to you class that there is molecule one. We have got also molecule two and molecule one. So we have got these molecules that are being formed, molecule 2 and molecule 1, which are identical. So in that respect, we can as well understand to say two identical DNA molecules are formed. Two identical DNA molecules are formed, as you can see there. They are formed, as we can see, class. Two identical DNA molecules are formed, which can be well understood that it is what? It is a DNA. Those are when two identical strands of DNA formed, they represent what? They actually represent DNA replication. Our next question, 2.3.2, identify monomer Y. Identify monomer Y. What is monomer Y? We can see Y, our Y there. I already have mentioned it already. Anyway, you can see these techniques class. I'm making sure that when you go to exam, make sure as well that you can be able to to pick and quickly mark them before you start writing. So then we cannot forget the answers. So it is what? It is the nucleotide. It is a nucleotide that is shown by Y there. You can really see class that is a nucleotide where we've got our nitrogen base, our phosphate, and our sugar molecules together there. And then identify the sugar in structure X, identify the sugar in structure X. Where is our X? Our X, which is the DNA structure. Identify the sugar in structure X. Our X is actually given that this is our X, which is representing a DNA structure. So what is actually just asked here is for you to know the type of sugar that is in what? In DNA. So it's deoxyribose. This is deoxyribose sugar class. Deoxyribose sugar is the one that we know that it is found what? On structure X, and then bond Z we can quickly see the nitrogen bases are attached together by a weak bond. What is the name of this bond? Actually, I'm quite sure that from our previous lecture again and lesson, this can be known, or this is known as what? As weak hydrogen bond. Weak hydrogen bond is actually given there. Weak hydrogen bond class. This is a weak hydrogen bond that is given there. Weak hydrogen bond is actually given there. Please take note of that. And then give two reasons why DNA replication is of biological significance. It's just actually asking us to say what is the importance of what? Of DNA replication. 2.2.3. We would need to put some. It's how many marks? 2 marks there. Just question 2.3.3 Importance of DNA replication. Importance of DNA replication. So we must understand the importance. One, we are saying one thing that you must understand here is that during mitosis, one daughter cell will divide into two identical daughter cells. During mitosis, one daughter cell will divide. It will divide into two identical daughter cells. Divide into two identical daughter cells. Two identical daughter cells. And then for each daughter cell to have identical genetic information as the mother cell DNA, is to make a copy before division. DNA replication must occur before division. Such that the daughter cell will match to the parent cell. It will match or it will be identical to the parent cell. That is one reason there that you must understand. We must understand that this is quite important, that before cell division or when mitosis takes place, remember that mitosis is the division of cells in order to ensure that they replace dead or worn out cells. So the cells that are being formed in mitosis, they must be identical to the ones that have been there before. So we are saying before the cell division, DNA replication has to take place in order to ensure that a replica of the cell that is going to be formed after the mitotic division is supposed to be formed, is going to be produced. That is one thing that we need to talk about, or we need to understand. Each daughter cell then contains the exact DNA composition as the mother cell. That is one thing that you must also understand. Each daughter cell must contain the exact DNA composition, must contain exact DNA, composition as the mother cell. As the mother cell class. That is quite straightforward. I mentioned and I have already explained. So tabulate two structural differences between mRNA and tRNA, messenger RNA and tRNA, transfer RNA. So the question wants us to tabulate. So it needs us to give a table that can just comparatively assess or analyze mRNA and tRNA. So we can give to say mRNA, and then we can draw a table there and say tRNA on the other side. So we can give to say tRNA and mRNA, they are given there as a table. So we can say one, what is the differences? One, mRNA is actually mRNA, we are saying it has got, mRNA is unlimited nucleotides or nitrogen bases. mRNA, it has unlimited, it has got unlimited or many, it has got unlimited or many nucleotides or nitrogen bases, many nucleotides or nitrogen bases. Which we can also give comparatively so to, which we can as well compare to this one. We can as well compare this. What's the difference with this one? With tRNA, only 3 nitrogen bases or 3 nucleotides is only 3. It has only 3 nitrogen bases. I'm sure that we can agree. We can properly agree. I can give an example to set mRNA is a single strand which have got many nitrogen bases that are going to be read by what? By this is our mRNA, and our tRNA, you can remember and reflect very well that it has got only what? It has got only 3 nitrogen bases. It only has 3 nitrogen bases, which is like that. So understand that there are only 3 nitrogen bases that are there, which we can say either we can say G, AGC as the nitrogen bases that are found there. Here we can give as many as we can. Here we can give them to be quite a lot of as many as we can say. There we can say it is G, C, T, A, C, A, T, G, C, T, A. You can see that there are many nitrogen bases that are found there, unlike in what? In tRNA, which has got only 3 nitrogen bases that we can see, that we have there. And then another difference is that in terms of this one, 3 nitrogen bases form a codon, and then 3 nitrogen bases form a what? An anticodon in terms of the tRNA, the mRNA and tRNA. So number 2, 3 nitrogen bases, 3 nitrogen bases, they form a codon in mRNA, and then in this side, 3 nitrogen bases, 3 nitrogen bases form anticodon. I'm sure this we can understand, and we can accept that we have got that as a different. We can also maybe want to give another third difference. There is also another third difference, which we can say that mRNA is linear structure and single strand is a linear structure, or is a linear structure. It has got a linear structure or a single strand. Single strand is found in this regard. And then if we look onto this one, if you can look onto this one on tRNA, it has got 3 nitrogen bases that form one codon. It is actually like saying that in this regard, it also has got what? A folded structure. In terms of the structural differences, it has a folded structure. It has got a folded structure. It has a folded structure in terms of the structural differences. This one is got a single linear structure, but in terms of the tRNA, it has got a folded structure. It has got a folded structure which forms, or which is a folded, or we can say a T structure. T structural shape. that can be quite a straightforward difference. So when they are saying table 8 class, you must draw a table where you can comparatively list the differences that are between the two items in question. So as you can see, we said mRNA on the other side and tRNA on the other side. But in terms of this, I was just drawing the structures. These structures, I'm drawing them only for class purposes, class. I'm drawing these only to make sure that I can be able to show you exactly what I'm trying to mean when I'm drawing or when I'm referring to these diagrams. You are not supposed to draw diagrams anyway. You are supposed to explain yourself. Myself, I'm just trying to draw the structures for you to see exactly what I'm referring to, what I mean when I'm talking about the differences in tRNA and mRNA. So understand these to say these are the differences that you must look at. And then next question is 2.5. The diagram represents one cell in a phase of meiosis. That's a one cell in a phase of meiosis. That's the diagram that is given there. The diagram that represents one cell in a phase of meiosis, that is what is given there. So let us quickly answer the question without also wasting more time for the interest of our time as well. So this is the diagram that is shown there. We can also see spindle fibers that are shown. And then let's quickly check what does the question want us to do. Identify the phase of meiosis illustrated in the diagram. Identify the phase of meiosis that is shown in the diagram. What phase of meiosis is shown there in the diagram? I'm sure, class, I told you that you cannot go to an exam and then you go to the exam without understanding meiosis and the specific sequential orders of this. This we cannot want to discuss anything more. This is what this is anaphase. This is anaphase 2. This is anaphase 2 that is shown there. It's anaphase 2 that is shown in this. Anaphase 2 that is shown in this. Identify the process that takes place at A. What is the process that takes place at A? You can see those are two chromosomes that are together there. They did not separate. So what is the process that where two chromosomes fail to separate? This is called what? Non-separation of chromosomes, or non is what is non-disjunction. It is, as it is most commonly known, is called non-disjunction. Non-disjunction, that is actually what is given there. And then if gamete B results, if gamete B results from a process identified in question 2.52 above, 2.52 is involved in fertilization with a normal gamete, explain how the zygote will be affected. Basically, what you are asked here is that you just need to understand the consequences of of non-separation of chromosomes. So what is supposed to be what you are supposed to state here is that one, in this question 2.5.3, we are saying one, the thing that you need to explain here is that the gamete will have one extra chromosome. Is this one results in fertilization with a normal one. The gamete, the gamete will have, it will have one extra chromosome. We have one extra chromosome. The gamete will have one extra chromosome. I'm sure that you know because it is not separated up there. The gamete will have one extra chromosome, which is actually like 7 chromosomes in this case. 7 chromosomes will be formed there. And then what other thing is that therefore the zygote will have an extra chromosome, which we said that in simple terms or in scientific explanation of non-disjunction, the gamete will therefore be like 2n. It will therefore be what? Be 2n plus 1, which is something which will result in what? Resulting in 13 chromosomes. In 13 chromosomes in the what? In the zygote when it fuses. Remember to say that gamete is before fertilization, which is formed. So in this case, it is the gamete, it is the zygote that is supposed to be having we have an extra 2n plus 1. It is the zygote that we have that number of chromosomes that we are referring to. Don't make mistakes, class. After fertilization, that's when we have 2n plus 1, but it is the gamete which is before fertilization, and then the zygote is after fertilization. So please take note of that. And then draw a label diagram of gamete B as it would appear in telophase 2. Draw a diagram of gamete B, which is this one we got. Draw a diagram of B as it would appear in what? As it would appear in telophase 2. So we can just draw a simple diagram quickly to see structure of gamete B during telophase. There should be, when you are asked to draw a diagram, always you must understand that you give the caption or you give the title 2.5.4, and then our caption structure of gamete B during telophase 2. Structure of gamete B during telophase 2. So that is your title of the diagram that you are asked to draw. So I'm just going to quickly draw the diagram. We can have our diagram there, which we can also have that is our cell membrane, of course, and then we also have our nuclear membrane that must be inside. We must have our nuclear membrane, and then we need to label them anyway later, but I can just say this is a what? This is the cell membrane. This is the cell membrane, and then, of course, this is the nuclear membrane. Nuclear membrane. This is the nuclear membrane, which is shown there. Cell membrane, which is shown there. Nuclear membrane, and then we have got, of course, our what? Our chromosomes that are given here. We have got our chromosomes that must be there, as to say that our chromosomes, and then we have got 1, 2, 3, 4. We also have this one must have its legs that side, and then here's the head, which also should have this. And then we have got our nucleus somewhere there. So as you can label this class, so we are saying nuclear membrane, cell membrane, and then we have got this as the nucleus. Is the nucleus. Nucleus is given there. And then we have got something like you need to understand to say here on top here, what is it representing? It is representing the what? The centromere. The centromere which joins the two chromatids together is given there. We also have we have got the chromatide, which must be just labeled as a single piece of it. The chromatide. My diagram is small anyway, but just take note. Chromatide is given. Obviously, the space which is there, which we are saying is this one, is the nuclear plasm. You must understand all these to say that they are what? They are quite important to be indicated on your diagram. So we are saying the diagram must be just easy, must be quite simple and well labeled. They must be there. So class, I'm sure that if I can just indicate this diagram, which I've shown that it has got the diagram such as that, I can also illustrate it to say that we have got a centromere which is holding the chromosomes together. That is illustrated by this diagram there. So thank you so much for this, and I'm hoping that whenever questions of meiosis comes, you won't be confused and you get them clear and straightforward. Let's just quickly answer, I guess, the last part of the question, which is alleles that determine the texture of human hair, and allele that results from what? That results in a curly hair, and an allele that results in straight hair. In a heterozygous condition, the hair texture will be wavy in phenotype. So that is it, straight hair, that is the allele, and then the wavy hair, and then the curly hair. We can see class this the diagrams that is shown. And then quickly, you must understand that the curly hair is represented by what? By dominant R. An allele that results in straight hair is represented by dominant T, as shown in there. So what is happening is that in a heterozygous condition, the hair texture will be wavy in the phenotype. It will be wavy in the phenotype in a heterozygous condition. So that is the what? This is the wave, and this is a heterozygous condition. It's a heterozygous condition that is given there. So what are we saying? What type of dominance occurs in hair texture? What type of dominance is occurring in hair texture? As you can see that when one allele completely covers or completely dominates the other, I'm sure that you know that type of dominance is called what? Complete dominance. It's called complete dominance because we are saying the hair can be straight, it can be curly. So when one of the alleles completely dominates the recessive allele, it is called what? It is called it's this one. In this case, we have got the formation of a heterozygous, which whereby to say a heterozygous is now there where we are having our wave here. So in this case, this is a what? An incomplete dominance. incomplete dominance is represented there. And then how many genes control the texture of hair? How many genes control the texture of hair? In this case, how many genes controls the texture of hair? We can quickly agree that there is only what? There is only one gene. It's only what? It's only one. Only one controls the texture of gene. It is given only only one gene controls the texture of hair. Only one gene class. Take note of that. And then Mr. Anderson has curly hair and Miss Anderson has wavy hair. Use a genetic cross to show the percentage of what? The percentage of possible phenotypes of Miss and Miss children. So in this case, this is quite an interesting question also. I remember I once presented it in paper in section A, where we calculated it. We say P1 phenotype, it must be given there. So we are saying Mr. Anderson is curly hair and Miss Anderson is got wavy hair. So we are saying curly hair, man, he marries a what? A wavy hair woman. Marries a wavy hair. Marries a wavy hair woman. And then we say genotype. Our genotype, we are saying the genes that represents that. So our curly hair, as it is represented in the question there, curly hair is represented by what? By R, which is that this curly hair is supposed to be what? Is supposed to be R, and then Mrs. Is what? Is wavy hair, which is a heterozygous condition, of this. So what is happening is that Mrs. It's an intermediate of that. So we have got RT because it's a combination of both of the alleles. It is a heterozygous that is giving us that gives us a wavy hair, and then we are continuing meiosis occurs, and then we need to have our gametes that are going to be formed here. We are going to have one R, and then on this side we are going to have RT, and then we have got what? We have got R and T, they are formed. And then as we cross them, there, we would see that this R, it crosses with this R, and then we are going to have what? R is this R and this R, this one, and that R there, and then this R, it comes and then it crosses with this what? With this T, and then we have got RT. This is our what? This is our genotype. And then our phenotype, this R represents what? Curly hair, one curly hair, is to one wavy hair. We can see that. So the question wants us to give what? Use a genetic cross to show the percentage of possible phenotypes, the percentages. So in terms of the percentage, we have got one curly hair, one wave. So in terms of the percentage, we are going to say percentage chance of what? Of curly hair is equals to chance factor of curly hair, is to say it is 1 over total ratio, which is what? Which is 1 over 1 plus 1, which is 1 over 2 times what? Times 100 over 1, and then is to 1 also over 2 times 100 over 1. So if we simplify there, we get what? Half of 100 is 50% Kelly, 50% Kelly. We're gonna have our 50% Kelly there. 50% Kelly is to 50% is to 50% wave here. You see. So that is how we can do it. So our answer is 50% is to 50% chance of Kelly and what? And wave here. But most of you are used to calculate this question as a what? Through a Punnett square. You are used to calculate this question through a Punnett square. Just to simplify it for you, just to make it easier for some of you who are used to Punnett square, I'm just gonna show you now, but the answers remains the same anyway. We have got our Punnett square there, and then we can say the father there is showing this side, and then we have got the mother also is supposed to be downwards. The father is also there. So the father is RR, and then the mother is RT, and then we can cross them to say R, and this R, we have got RR, this R and that R there, we have got RR, and then this T plus that T, we have got RT, and then this T from this R, we have got RT again. So if you can look at this, you can also further simplify that. We have got two Kelly here as to two wave here, which we can say it is 2s to 2, and then we can simplify by dividing by 2. We can divide by 2, and then we get 1s to 1. 1s to 1, same as what we got above, and then which is also the same as what is 1 over 2 times 100 over 1 is to 1 over 2 times 100 over 1, which can also give us the same answer as 50% is to what? Is to 50%. So you can see, class, these answers, they are quite, they are weighted the same way. You can go up to the position of getting the gametes. You can go up to the position of gametes, where from this position, you can put it on a Punnett square, or you can do it as a genetic cross, as I've just shown you there. So please take note and don't get confused. Maybe last question. The last question, it is the table contains diagrams representing the body parts of different primate species, A, B, C, D, E, F. Which diagram represents the pelvis of a pedal organism? The pelvis is must for a pedal for the pedal organism. It must be what? Be shorty, upper. It must be upper and broady, or wider, which can be what? Which can be C. So our answer in this case, it is C, and then give one reason for your answer. I mean, this reason I already mentioned it, but let me just write it again, that the pelvis is short and wide. This question 2.7, it has got the pelvis is up, short, and what? And wide. That we can say in terms of pelvis. In terms of pelvis, see, it is shorty, it is up, and it is also broad. As you can see there, it's broad. So we are saying the answer is what? The answer is C, because of that reason. And then what type of grip is possible due to the structure of part F? Part F, it is just giving the what? The end, which has got the opposable thumb. So what is supposed to be understood in this case is that there is what? There is a precision grip or power. Precision grip or power is given in that question. And then because of the opposable thumb, and then give all the letters of the body parts that belong to a chimpanzee. A chimpanzee, we can say it chimpanzee, all the body parts that can represent a chimpanzee. We can have A can be okay, B cannot be, C cannot be, and then we can also have D as well as what? As E, which is also a quadrupedal. So we are saying it should be A, D, E, A, D, E, A, D, E. They are the letters that represents the correct answer. Describe one difference between the teeth of organism B and D. The difference between D and D teeth. It is between B and D. Between B and D, the difference between B and D, which is B and D, what's the difference between the two in terms of their teeth? So here, one organism B has smaller teeth, as we can see. Organism B has got smaller teeth, that is correct, which we can see there. Organism B yes, smaller teeth, which can also be seen, which we can say they are what? They are canines, smaller canine, and D has got large ones. And D is large canines that we can see. And then other difference that we can see here, another difference that we can see also is that because organism A, it has got no diastema, and organism D, it has got, if you can check in terms of the teeth in A and B and D. So organism B has no, it has no, organism B has no diastema, and D it has. So that is quite another difference that we can see. So like I have said, I actually divided our paper into section B into different parts, question 3 and question 2. So this is the end of question 2, just to save you from long presentations. So we just did question 2 now. Thank you very much for watching, and look forward for your look forward for your last part of the question, which is question 3. It is just going to follow in the next presentation. Thank you very much for watching, and all the best in your exams. Have a good day.